my pleasure to be here today, and thank you so much for asking me to present at this wonderful symposium. I've learned a lot of exciting things, but I think my favourite fact so far is that hyenas really like watermelons. Who knew? Anyway, I'm going to be taking a bit of a step back, and we're going to look at things from a much sort of a wider point of view, perhaps. But my first slide is, you know, I think David Attenborough has it wrong. It's not a blue planet, and I think I'm in the right place to be able to say this. It's a green planet that we live on, and plants are probably one of the most important kingdoms that are present on Earth. Forests on their own make up 31% of Earth's land surfaces. And this is my favorite factoid of the last couple of years. I think it's from a science paper last year. Of the 550 gigatons of carbon on Earth, 450 gigatons of that carbon is plants, or comes from plants. Okay, so just to contextualize that, in case you're having trouble visualizing what 450 gigatons of carbon looks like, that's the same mass of carbon as 90 billion African elephants. Also difficult to visualize, but you know, you get my gist. Plants are really important. It's a green planet that we live on. But it wasn't always like this, okay? If we go back 500 million years, Earth was barren. There were no plants on its terrestrial surfaces. Might have looked a bit like this, so no soil, no greenery to speak of. And at the water's edge, things were a bit more exciting, maybe. There was some algal sort of biocrust and algal masses, cyanobacterial mats growing at the water's edge, but no real plants to speak of. But then something amazing happened about 500 million years, and plants made a transition from being aquatic organisms to becoming terrestrial. And they managed that without roots, okay? So how did they do that? And this is a question that people have been asking for many, many years. And we can get some clues as to how they might have done that from fossils, okay? And these fossils are amazing, okay? These are about 412 million years old, and you can quite clearly see they're plants. But they're some of the earliest plants that came onto the land. And actually, in real life, they're about this big, so they're tiny, tiny. And you've got branching stems and little spore bodies on top of these stems that branch. But you'll notice neither of them have got roots. Okay, and why is that so important? Okay, plants normally get nutrients from the ground, from the soil, through their roots. Okay, but these plants didn't have any yet. They were able to get onto the land and live in this barren, soilless, terrestrial environment without these roots. But luckily for us, we can look to fossils and we can get clues as to how they might have done that. So this is my really poor drawing of one of those fossils. This is Aglaophyton major. And the fossils that we're able to get from this time period, some of them are really amazing. And some of the most amazing fossils from that period come from something called the Rhynie Chert. Okay, Rhynie is this tiny village in Scotland, but back in the sort of when plants were sort of making their moves onto land, Rhynie was a very different environment to what it is today, and it was like this wonderful sort of thermal terrace environment with very highly silicified waters, and things were falling in the water and getting preserved everywhere. It was amazing. But this Aglaophyton major fell into these highly silicified waters and absorbed those minerals and became a fossil. And these fossils are super amazing. I think I've said amazing enough times now to convince you, but in case you're not convinced, this is a slice through one of those fossils of the axis that sort of snaked along the ground, we think, of Aglaophyton major. And you can see the preservation in this fossil is such, you can make out individual cells within that axis, okay? Pretty incredible, yes? More incredible, if you look more closely at those cells, particularly in this band around the outside of the axis, you can make out intracellular structures within those, okay? And these structures, they look pretty familiar if you're someone like me and you look at plant roots for a living. They look very similar to these ones, which are stained from a root from a wheat plant just a few weeks ago. So we've got these highly branched structures, which look a lot like this. We've also got these vesicle structures and these hyphae that run between the cells, and you can see all of these structures in these fossils. So this makes us think that those early plants formed partnerships with ancient fungi, and that these ancient fungi were very similar to the ones that form partnerships with plant roots today. What are these partnerships called? We call them mycorrhizas, okay? Mycorrhizas are formed between fungi that live in the soil and plant roots, in the most part. We call them mycorrhiza-like in plants that don't have roots. The spores germinate in the soil, so this is some spores we have in culture germinating, and they come across plant roots germinating in response to signals that the plant roots are releasing into the rhizosphere, and they form contact, and then they branch within the roots, forming those characteristic structures that I just showed you earlier. So these highly branched ones we call arbuscules, and then these swollen fungal vesicle-like structures. Now, what do they do? We normally think of mycorrhizas as being mutualistic partnerships, okay? So plants fix carbon dioxide from the atmosphere through photosynthesis and produce a wealth of carbon compounds, which they then pass to their fungal partners through these arbuscules or vesicles within the root system. In return, the fungi get nutrients out of the soil and transfer them to the plant root system, which then gets assimilated into the green parts of the plants and used in their metabolism and various processes within the plant itself. And so in this way, you get this bidirectional exchange of carbon for nutrients between the plant and the fungus. And that makes us think, well, that makes us appreciate their mutualistic nature. Now, in truth, mycorrhizal symbiosis, whilst it's normally considered to be mutualistic, it can actually lie anywhere on this continuum, all right? So on the one end, you've got these mutualists, where you've got give and take, but on the other end, you have parasites, okay? You have some plants that are entirely dependent on their fungal partners for everything. They sponge carbon out of them, they take the nutrients, and they don't give anything back. And you have some that sort of oscillate in between, things like orchids, which germinate with a fungal partner, take all they can get, but then they pay it back later on in their life cycle once they start photosynthesizing. And we know where these symbiotic partnerships lie can be anywhere on this continuum, and that where they actually lie on that continuum is affected by a number of different factors, including things like competition, the genetic diversity of the plants in question, compatibility between the plant and the fungus, and also environmental factors like soil nutrient status, soil types, and CO2. So the CO2 thing is really important when we start thinking about how plants go onto the land and the role of mycorrhizal fungi, because when plants are making that transition from an aquatic to terrestrial existence, CO2 in the atmosphere was nearly 10 times what it is today. And this will have affected how those mycorrhizas were functioning in those early plants. So we know modern angiosperms at this end of the scale, evolving a mere 130 million years ago. We know in most cases they are mutualistic in these plants. But a lot of the plants earlier on in the plant phylogeny, we didn't know what they did until quite recently, where we carried out a bunch of experiments to look at carbon for nutrient exchange at different CO2 concentrations, replicating paleo-atmospheres of about 1,500 parts per million and modern-day CO2, and trying to identify whether it was mutualistic or not. And the great news is, whilst we know in flowering plants it is mutualistic, but also in all the other clades, we found that they were also mutualistic, and they were mutualistic at both high CO2 and low CO2. So this gave us some more evidence to suggest that actually mycorrhizas were key in helping plants make that transition from an aquatic to terrestrial lifestyle. So today, we'll just zoom forward 400 million years. Today, more than 90% of plants have these fungal symbionts in their roots. And most of the time when you look at a plant, it's not just a plant you're looking at a plant and a fungus. And that includes many of the crops that we rely on. So in the wild, we've shown previously that mycorrhizas actually help plants get nutrients out of the soil from sources beyond the root depletion zone, but also from alternative sources in the soil. So things like from rocks or from organic substrates. They also help enhance plant defense against disease and pests by priming the plant defense system. They can also help increase plant drought tolerance and resistance to drought, although the mechanisms for that are a bit unclear still. So our challenge then is to take all this knowledge that we've discovered by using wild plants and translate it into crops against this background of global climate change. So instead of looking to the past, where we're looking at high CO2, we now want to look at the future and what the future responses of these mycorrhizal partnerships are going to be as CO2 rises into the future. And this is kind of the underpinning question that drives a lot of the research that we're doing today, and that is, can we make the most of the soil fungi to help us feed the world in the future? Now again, I'm sure I don't really need to spell this out to this audience, but we're all aware that one of the main challenges facing humanity is food security, and particularly future food security. So we know agricultural productivity has increased rapidly since the Green Revolution of the 1950s. And this figure here just shows the wheat yield since the 1980s. And you can see it steadily increased, and then it's starting to plateau out around the late 90s, early 2000s. This steady increase in wheat yield has largely been down to the development of novel chemical pesticides, fertilizers, big leaps forward in plant breeding. But we've now reached the state where the crops aren't really becoming as responsive to increasing fertilizer use. And increasing usage of those fertilizers is totally unsustainable anyway, for reasons that we heard earlier this morning. So this depletion of natural resources and sort of the increasing human population means we're creating this perfect sort of storm, and we're facing this food security crisis in the future. We don't have the resources to keep producing more fertilizers. They're energetically expensive, they're monetarily expensive, and they're also finite. A lot of the raw materials used to make them are finite. And so we want to know, maybe mycorrhizal fungi could be the sustainable solution to this. We know that wheat and barley, for instance, are capable of forming these mycorrhizal associations, but we also know that ability to form those associations is highly variable. And this is because breeding for different characteristics actually inadvertently selects against being mycorrhizal. So things like responsiveness to fertilizers, disease resistance, fungal pathogen resistance, you're actually selecting against being mycorrhizal by selecting for those traits. But what we don't know really, we don't have a firm handle on, is how variable modern wheat varieties of wheat in particular are to mycorrhizal colonization. We've also shown through previous work that in wild plants, colonization and function is dramatically affected by CO2 concentrations. We know CO2 is rising in the future. So not only have we got these huge knowledge gaps that we don't know how well domesticated crops respond to mycorrhizas, we don't know how well the mycorrhizas themselves respond to increasing CO2. So we've got this double whammy of issues. So we've conducted a bunch of experiments back in Leeds, looking firstly at mycorrhizal receptivity, and this is the work of one of my PhD students, Ashley Elliott, which is currently in review, hopefully, for Plants, People, Planet, where we grew 3 different cultivars of wheat. And these are elite cultivars that are currently routinely used in the UK. We grew them in a non-sterile soil mixed with sand. And this soil we collected from the Leeds University farm, so it's agriculturally relevant. And we then either supplemented that soil with or without an inoculum for mycorrhizal fungi. We then assessed the amount of mycorrhizal colonization in the roots after 8 weeks of growth through simple root staining and microscopy. And what we found is quite interesting, I think. So this top figure is the only one I really want to draw your attention to, which is how much of the root is colonized by fungus with and without the inoculum added. So if you look at the black bars, this is where we didn't add any inoculum, so it's just the wild agricultural mycorrhizal community infecting the roots. You can see there's big variations between the different wheat cultivars, with Avalon particularly highly colonized and Cadenza being the least colonized. But all 3 of them responded well to adding mycorrhizal inoculum with increased colonization of the roots. So that's great. We can increase colonization by adding mycorrhizal spores to the soil. But what does that mean? What does that actually do? Colonization doesn't mean anything, really, when you want to look at function. What do they actually do? So this is the work of one of my postdocs, Tom Thurk, who's currently in revision, and he conducted some isotope tracer experiments where he actually measured the amount of nutrients being assimilated by the plants via the fungal partner, and then the amount of carbon the plant was transferring to the plant in return. So, and he did this at 2 CO2, so we did this at modern CO2 and we projected future CO2 atmosphere. And what he found, this is total phosphorus, so this is plant phosphorus and mycorrhizal phosphorus together. He found those differences between the cultivars. I don't know why the colours haven't come out, but this one here is modern CO2 and this one is the future CO2. And there were different responses to an elevated CO2 atmosphere. But how much of that was thanks to mycorrhizas? And this is what he found. So he had a huge variation between cultivars in how well the mycorrhizas were functioning in those roots of those different plants. So ambient CO2, actually, we got more well, there was no statistical difference, but the trend was for there to be more phosphorus from mycorrhizas in Avalon and Cadenza. But Skyfall, the opposite seemed to be true. You got more mycorrhizal acquired phosphorus at the elevated CO2. But most interestingly was this huge difference between cultivars. So we have one cultivar that's able to get a lot more out of its mycorrhizal fungal partners than the others. Nitrogen, we also did this for nitrogen. And again, big differences between cultivars in terms of total nitrogen. We also get this nitrogen dilution effect of bigger plants having lower concentrations of tissue nitrogen in their above ground tissues. and mycorrhizal acquired nitrogen. There was no difference between CO2 concentrations, but differences between cultivars. In terms of carbon invested, was not affected by CO2, and that was super interesting because that actually decouples carbon from nutrient exchange in the mycorrhizal symbiosis in wheat. So just to summarise, it was a super whirlwind tour of stuff that we've been doing in Leeds. We found this cultivar-specific receptivity to mycorrhizal colonisation, and that's probably a result of different of intensive breeding for different traits that are actually selecting mycorrhizas. We found that mycorrhizal fungi can contribute to wheat nitrogen and phosphorus uptake, and that this is variable according to cultivar. We've also found that the nitrogen and phosphorus uptake via mycorrhizal fungi is not linked to plant carbon outlay, which goes against a lot of the mycorrhizal literature. And also that this symbiotic function is not affected by atmospheric CO2. So overall, it's a good news story. Mycorrhizal fungi have a potential role in helping us develop sustainable approaches to future agriculture. And that's that. Thank you very much. So thank you to all my group and also my collaborators and funders on this side. Thank you.